Welcome to Biology at Priyajit, my dear viewers. Today we explored an interesting aspect of echinoderm biology, that is larval forms of echinodermata. These larvae are bilaterally symmetrical and free swimming, strikingly different from the adult body plan. So studying their structure and types help us to understand echinoderm development, evolution and metamorphosis. Let's take a brief look at these fascinating transitional stages in the life cycle of echinoderms. So this is the image showing the bipinnaria larva, brachyolaria larva, auricularia larva and doliolaria larva. There are different larval forms of echinoderms. So now we should study in detail. Why we study the larval forms of different echinoderms? Studying the larval forms of different echinoderms is very much crucial for various ecological and scientific reasons. First one is the evolutionary insights. Larval forms reveal important clues about evolutionary relationships among echinoderms, highlighting both conserved and divergent traits during development. Developmental biology. Larval forms help explain embryogenesis, organogenesis, and metamorphosis, while highlighting the transition from planktonic to benthic life. Phylogenetics and systematics. Larval structures and developmental patterns also help in constructing phylogenetic trees and understanding ancestral traits. Ecological importance. Larvae aid or help in dispersal and colonization, maintaining genetic diversity and influencing population dynamics and resilience. Life history strategies. Larvae provide insights into the reproductive strategies such as this balance between planktonic dispersal and benthic settlement. Biometric application. Studying echinoderm larvae inspires biometric designs in engineering technology such as ciliary-based locomotion. Paleontology and fossil records. Larval features help link extinct echinoderms to their fossil ancestors, bridging gap in the evolutionary timeline. So larval forms in phylum Echinodermata. In echinoderms, egg and sperms are released in water and fertilization takes place in water forming zygote. Echinoderms are deuterostomes and hence their cleavage is very much is radial, holoblastic and indeterminate type. So the larva hatch in water and feed and grow through successive larval stages to become adults. And the larvae of echinoderms are bilaterally symmetrical, but they lost symmetry during metamorphosis. Different classes of echinoderms show structurally different larval stages, and their comparisons can reveal their evolutionary ancestry. Diplurula larva, hypothetical ancestral larva, basis for all echinoderm larval types. Symmetry, by the larvae are bilaterally symmetrical unlike the radial adult form. Structure, their oval body, it has ciliated epithelium, ventral mouth and posterior anus. Ciliary bands, it aids in swimming and feeding by capturing food particles. Digestive system, it is simple, complete with a straight gut. Locomotion, free swimming via ciliary movement. Significance, it shows evolutionary links among echinoderms and other deuterostomes. Development, the larval forms of echinoderms evolves into specialized larval forms and their metamorphosis into radial juvenile. Pentactula larva. This is the image of the pentactula larva. This is the final larval stage of Holothuroidea transitioning to adult form. So its symmetry, it shows pentaradial symmetry like adult sea cucumbers. Structure, they are small, cylindrical with 5 tentacles around the mouth and tube feet for attachment. Ciliary bands, they reduced compared to earlier larval stages. Digestive system, they have functional gut, begins benthic feeding. Locomotion, tube feet aid in crawling and substrate attachment. Development, it transforms into a juvenile adapting adult features. Significance, this is the key stage in shifting from planktonic to benthic life cycle. This is the significance of this pentactula larva of Holothuroidea. Bipinnerial, this is the pencil sketch of bipinnerial larva. They are early free-swimming larval stages of asteroidea or starfish. Symmetry, they are bilaterally symmetrical unlike the radial adult form. Their structure, they have oval body with ciliated bands for locomotion, lack skeletal structures or arms. Ciliary bands, they have complex forming loops for swimming and feeding on plankton. Their digestive system, functional gut with a mouth, esophagus, stomach and anus. Locomotion, it moves using ciliary action. Development progresses to the brachylaria larva, grows additional arms and adhesive structures before metamorphosis. Significant, it represents the initial feeding stage in starfish development and a link to evolutionary adaptations. Brachylaria larva, this is the pencil sketch of brachylaria larva, a later larval stage of asteroidea or starfish developing from the bipinnaria larva. Symmetry, bilaterally symmetrical, similar to bipinnaria. Structure, elongated body with ciliated bands for locomotion. It features additional arm-like outgrowths called brachyolarial arms. Arms are equipped with adhesive structures for attachment. They have a preoral lobe is prominent. Ciliary bands, they are well developed structure and aid in swimming and feeding. Digestive system, their fully functional gut which is similar to bipinnaria with a mouth, stomach and anus. Locomotion, it uses ciliary bands for swimming but begins adapting for a benthic lifestyle. Their development, it prepares for metamorphosis into the juvenile starfish and transitions from planktonic to benthic stage like pentactular larva. Their significance represents an intermediate stage in starfish development, showing adaptations for a sedentary adult life. Auricularia larva, this is the pencil sketch of auricularia larva, which is the early free-swimming larval stage of Holothuroidea or sea cucumbers. Symmetry, they are also bilaterally symmetrical, unlike the radial symmetry of adult echinoderms. Structure, there is transparent oval body with a distinct ciliated epithelium. They features a prominent circular ciliary band and additional transverse bands. Simple digestive system with a mouth, gut and anus. Ciliary bands, they have well developed and ciliary glands which are used for locomotion and feeding. It helps capture planktonic food particles. Their locomotion moves efficiently through ciliary action in planktonic habit. Digestive system, they are functional with the ability to ingest and digest plankton. Development, grows into the Doliolaria larva as it advances in development, begins exhibiting features preparing for metamorphosis. Significance, it plays a crucial role in dispersal and planktonic feeding during the holothurial life cycle. Doliolaria larva, which is very important, is the pencil sketch of Doliolaria larva. It is a transitional larval stage of Holothuroidea, sea cucumber and crinoidea or sea lilies, evolving from the auricularia larva. Symmetry, it retains bilateral symmetry but begins showing features of pentaradial symmetry. Structure, barrel-shaped body with transverse ciliary bands for locomotion. Ciliary bands, they are reduced and organized into distinct rings. Digestive system, they have functional gut. Their feeding activity decreases. Locomotion, it moves through the ciliary action but shows reduced mobility compared to auricularia. Development, it transforms into pentactular larva preparing for a benthic lifestyle. Significance, their key stage in transitioning from planktonic to benthic existence. Pluteus larva, this is the Ophiopluteus larva and Echinopluteus or Pluteus larva of Ophionotus hexacticus. Pluteus larva, they are free-swimming larvae of Echinoidea or sea urchins and Ophiuroidea or brittle stars. So their structures, they are transparent T-shaped body with ciliated arms and calcareous rods. The ciliary bands which help in locomotion and planktonic feeding. Digestive system, they have functional gut which feeds on plankton. Locomotion, it uses ciliated arms for movement. Development, Echinoidea, it becomes juvenile sea urchin. Ophiuroidea, metamorphosis into juvenile brittle star. So the significance is the key for dispersal, feeding and early survival. Antedon or yolk larva, their structure include ovoid or spherical transparent body with a yolk sac for nourishment. Ciliary bands of a cilia aid in locomotion and feeding by generating water currents. Digestive system, which is rudimentary, depends on yolk reserves during the free swimming stage. Locomotion include free swimming driven by ciliary action for movement. Development undergoes metamorphosis, attaches to a substratum and transforms into a sessile pentacrinoid larva before maturing into an adult. Significance: it is essential for dispersal and exhibits symmetry and plasticity typical of Echinoderm larvae. Cystidian or pentacrinoid larva, this is the image of the pentacrinoid larva. It is small stalked larva with a sac-like body attached to a substrate. Ciliary bands, they are less prominent, aiding in minor water movement. Digestive system is more developed, begins feeding as yolk reserves deplete. Locomotion, sessile anchored to the substrate via a stalk. Development, it is a transitional stage, developing arms and feeding structures. Significance, it marks the shift to a benthic lifestyle and adult echinoid formation. So this is the image of different types of echinoderms. So as we conclude, remember that larval forms of echinoderms provide vital insights into their development, metamorphosis and evolutionary history. Their bilateral symmetry and gradual transformation into radially symmetrical adults highlight key biological transformations. Understanding these larvae is not only important for exams but also helps us appreciate how complex body plans emerge during development. Keep revising, stay curious and continue exploring the wonders of animal life cycles. So thank you my dear students.